Hello, my name is Franco Amaro and I'm going to present algorithms and cancer research. Networks of gene interactions in which gene activates and represses the transcription of other genes are responsible for much of the complexity of cellular life. We know too little about how genes are created or transformed. Cancer research is taking place with a lot of new technology, this involving interferential statistics. The input gene expression data will be assumed to be the statistic single-cell data, unless otherwise noted. This is meaning that we are measuring only with one cell and with one time point of data. For the problem statement, we know that cancer arises for a loss of regulation of processes into cells, and understanding disease structures is going to be the underlying networks and powerful insight into the discovery of relevant genes. For convenience, it can be useful to abstract the problem of network interference into a graph theory framework, as we can see in Figure 1. Given the large number of genes measured in these experiments, it may be prudent to select a smaller subset of genes with high biological variance for further analysis. This for having the statistics going with the way we want and with the measures we want. For the results, we have this correlation algorithm class, regression, Bayesian simple, Bayesian dynamic, informational theory, and Pfizer. This is how we are going to measure our results of cancer research and the statistical ways to do it. One of the most basic methods of network interference is to compute the correlation of each pair of genes. Regression-based algorithms are designed to both linear and predefined nonlinear interactions. Bayesian methods are an interaction between genes and are represented to the conditional probability that we can see and quantify. Informational theory is a theoretical framework for quantifying information and studies in its properties. Pfizer has the advantage of producing an output graph that is both directed and can contain cycles. As this, we can see that most of algorithms has disadvantages and advantages. Correlation algorithms are fast, scalable, and have a good detection of forward loops. Regression has a good overall accuracy. Bayesian simples and dynamics perform on small networks and also in the detection of cycles. Information theory has detection of feed-forward loops, fan-ins and fans-out, similar to correlation methods, but with much more accuracy. And Pfizer is an output that has a good setup and is also available for research. For the discussion, it has been seen that it has long been thought that combining an assortment of different algorithms could prove beneficial into network interference. An interesting current problem is how best to combine information from multiple data sources in order to generate a prediction the best way. There have been so many exciting new applications of network interference that we cannot provide in a depth explanation of all of them. This is because of the complexity of genes and human genome. And for conclusions, research is becoming a new advanced tool to work with, and we can find out new solutions and projections, in this case for cancer research and new involvements of technology. We can now discuss about the basics of network interference, different types of algorithms, and some unsolved computational challenges, and new exciting applications in cancer research.